Thank you, and welcome to this morning meeting in the US and afternoon meeting in Europe. My name is Sven-Erik Dahlén. I'm professor of asthma and allergy research at the Karolinska Institute in Stockholm, Sweden. I do this presentation because I'm a member of the FreeTR team, and I have got the task to introduce this meeting and to give you some background about what FreeTR is. So I'm site leader of the FreeTR project at the Karolinska. As you can see from this busy slide, there are 70 partners in this unique and largest ever IMI project to be started about 1.5 years ago. I'm involved in the steering committee of FreeTR because I'm co-lead of the activities in asthma and COPD. But I'm also lead of the Workpackage 10 that actually is the partner of this c-path activity event today. So what is then FreeTR? It's really the ultimate precision medicine project that attempts to do something that has never been done before, trying to find commonalities and differences between 7 immune-related diseases, that is rheumatoid arthritis, systemic lupus erythematosus, multiple sclerosis, the 2 inflammatory bowel diseases, ulcerative colitis and morbus Crohn, and then asthma and COPD. And as I said, this project started about 1.5 years ago, and it has a funding of 40 million euros in public money, and with in-kind support of the equal amount from the industry. So I thought for some of you, this IMI is probably new, whereas others know a lot about it. But just to remind you, the IMI project started more than 10 years ago in 2010. And it was really started to improve drug development in Europe, to try to improve the science to support innovative new treatments. And the money that was put on into this has been, as I will come to, very large, and it has been actually very successful, because the whole idea was to break down the barriers for collaboration between different disciplines, and in particular between academic scientists and industry scientists. So through support to these new collaborations, a lot of new model development has been supported, new standardization, implementation of new strategies, and specific research on a lot of projects, including what will be dealt with today, the impact on regulatory practice. And a particular focus of IMI has really been to involve patients in these different projects. Today, more than 100, actually 118 projects have been concluded, and altogether more than 5 billion euros has been invested during the lifetime of the IMI projects. And this is a remarkable success. I should mention that it's a very unusual type of competition for these grants, at least according to European standards. So it's a complicated process for the call and for the applications that I won't go into, but the bottom line is that winner takes all. So in all of the activities, there's only one of the competing consortia that wins. And I think this is one of the reasons why excellence has been very high. And as you can see from the figures here, a lot of papers have been published, and a significant share of them in the top cited papers. So that's really the background of what IMI is. It's a collaboration between public money and private money coming from the different pharma companies in particular, but also SMEs, small, medium-sized enterprises are and have been involved a lot in IMI. So coming back now to 3TR, the 3 T's stand for taxonomy, treatments and targets, and R for remission. And the overall winner that won this call was my colleague Marta Alcaron Riquelme from Granada in Spain. She is the academic lead and the PI of the whole project. The industry lead is Terry Means of Sanofi in Boston in the US. And again, as already shown on the first slide, the whole idea with 3TR is to try to find precision medicine biomarker panels across these 7 diseases, and to relate biomarkers that are really the core of precision medicine also to conventional and new clinical outcomes, and again with a particular emphasis on patient-related outcomes to different intervention responses. So the goal of 3TR is really to develop new and universal cross-disease definitions of response, remission, relapse, and resistance to treatment with different immune modifiers. So it's a very ambitious project, and as I think I mentioned, the project runs for 7 years, which gives it a, I think, reasonable chance to at least be successful for some of the very many visions of the project. So all these huge multi-partner, multi-stakeholder, multi-discipline projects need very firm organization to be successful. And 3TR is divided into 10 different work packages. Some of them, like work package 1 to 3, are more for the cohesion, for the management, and so forth. But if we look at the left side of this graph, the work package 6, 7, and 8 are really the core of the projects. That's the work packages where centers of excellence in the different disease areas recruit patients, examine patients, collect samples for analysis. These samples are then being analyzed in the profiling work package, and there will be integration between all these diseases in the work package 5 for disease activities. So one really central part is the biomarkers, and in the disease-specific biomarkers, what already is known will be used for the different studies, but there will also be interaction between the different disease areas to find commonalities, to understand what is similar, what is dissimilar between these diseases. And that's a very novel aspect of this project. Then in the work package 4, which is an overriding work package, the analysis will be further developed and added. And just to give you an idea, I mean, the different levels of biomarkers that will be used for the molecular phenotyping are ranging from the genome, the transcriptome, the proteins that are being translated into the actual molecules that carry out a lot of these messengers in health and disease in the metabolome. And this will be related to clinical appearance, response to treatment. And it's hoped that we will be able to define then different fingerprints, different handprints for the different diseases in this projects. Just to give you an idea of how material will be collected in FreeTR, one can say that we have introduced the carousel model. As you know, you can jump on a carousel by going to the elephant or to the donkey or the lion or whatever animal is going around in the carousel. And the same will be for sampling and getting experience and knowledge from different studies. That if we start at the top, FreeTR will certainly set up a few completely new studies within the different diseases, and hopefully also a joint study across the 7 diseases. But a lot of samples and analysis will also be carried out in studies that already had started before FreeTR, and then new samples can be added to ongoing trials. Furthermore, at the bottom, you see that there are clinical trials that have been completed, but where there is a biobank of samples that can be used for analysis within FreeTR. And then finally, the left box here, there are trials that have been completed where biological material has been analyzed, but the data can be shared with FreeTR. And FreeTR will very much use advanced bioinformatics to analyze this information that can come from the different type of studies. Now to today's activities, which is really something that Work Package 10 has been engaged in. I'm together with Takeda having the lead of Work Package 10, which actually is a very small work package, but where the objective is to try to provide a platform for communication with the regulatory authorities about biomarkers and personalized medicine. This is certainly not new. This is something that has been discussed for a long time. It's a fundamental part of the personalized medicine process to have predictive biomarkers that can be used for qualification in regulatory trials, and what can be used in clinical practice also. So the aims of Work Package 10 is really to create a platform for communication and exchange of experiences, and to define future activities and goals during this altogether 7-year period. So we want to learn what the regulatory authorities require, what their input and requests are, and we'll try to create an interface with the scientists in FreeTR to see what we can supply in this exchange. And with time, we wish to establish a regulatory working group within FreeTR with representatives of all the 7 diseases, and also a multi-stakeholder group, including patient involvement, where these things can be where we can be more conclusive, and at the end of the project, so to say, deliver a product that can be useful for all involved. So Initially, it has been I, myself, and my co-worker, Associate Professor Lars Andersson, that has tried to get this process started together with colleagues at Takeda, where Jiri Aubrecht initially was very active. He has now left the company. But Antonella is the driving force together with Akiki and Elizabeth. And my final slide is really that now today, we hope to learn from this regulatory workshop in collaboration with c-path. And then we have a plan to take this further into the FreeTR project, and we look forward to collaborations with patients and the regulatory authorities to improve personalized medicine. And today, the model will be of obviously inflammatory bowel disease. And with that, I end. Thank you.